Assalamualaikum warahmatullahi wabarakatuh. In this case study, we are from AS12050. We will be presenting high performance liquid chromatography to determine the caffeine contents in tea and coffee. High-performance liquid chromatography, or known as a HPLC, is a type of chromatography that employs a liquid mobile phase and a very finely divided stationary phase, and it requires a pump where it forces a solvent through a column under a high pressure. HPLC is quite same in terms of its operation with the liquid chromatography, but this allows far better separation of the component. This advanced chromatography enables researchers and scientists to analyze, quantify, and understand complex substances, making it indispensable in fields like pharmaceuticals, environmental monitoring, food safety, and more. In this case study, by using of HPLC techniques and with a given chromatogram of caffeine, the concentration can be generated and accurately calculated. This analysis not only illustrates the practical of utility HPLC in routine laboratory work, but also highlights its role in ensuring consistency and safety in consumer products. So let's dive into the methodology and result to explore how HPLC make this possible.
Next, we will move to the objective. In this experiment, we discovered two objectives. The first one is to determine the caffeine contained in tea and coffee, and the second one is to determine the retention time of a solution, which is the caffeine.
Moving on to the next slide. This schematic diagram demonstrates the major components and the operational workflow of a high-performance liquid chromatography HPLC system. We begin with the solvent surveyor, which stores the mobile phase. This mobile phase is crucial because it carries the sample through the system. The pump then takes over by generating the high pressure needed to push the mobile phase from the solvent surveyor to out the system. A consistent flow of solvent is essential for efficient separation of the sample components. Next, the injector, which introduces the sample into the mobile phase. The sample is injected carefully to ensure proper mixing with the mobile phase. Before entering the main analytical column, this sample passes through the guard column. This small yet important component is responsible for trapping any impurities or contaminants that could damage the main column. It helps extend the life of the analytical column by keeping it clean. The sample then enters the analytical column, which is the heart of the separation processes. Inside this column, the components of the sample interact with the stationary phase, and based on their chemical properties, the components are separated. Each component will have a different retention time based on its interaction with the stationary phase. As the separated compound exit the column, they are detected by the detector. The detector measures the compound as they issue from the column and passes the information to the data system. Finally, the data system and display will display the result in the form of chromatogram, where we can observe the retention time and the concentration of each separated component. The chromatogram gives us a visualized presentation of the separation processes. In short, HPLC operates by pushing a liquid mobile phase through a column where the sample is separated into its components, and these components are then detected and displayed as a chromatogram.
Here, the left one is the result of chromatogram of caffeine, and the right one is the result of chromatogram of caffeine in tea. So based on these two graph chromatogram, we determine that both has the same retention time, which is 2.6 minutes, but both have different peak area. For standard caffeine, peak area is 9 MV minutes, and for caffeine in tea is 4.95 MV minutes. So, to determine the caffeine content in tea and coffee, we look at the calibration curve equation, which is y equal to 25080x minus 1745.2, and then let the y as peak area as we found in previous slide and X is the concentration in ppm. So here we want to know the value of X and we should add the value of Y in the equation. For caffeine in coffee sample, the result is 0.0699 ppm, while for caffeine in tea sample, the result is 0.0697 ppm. So based on this result, we discover that coffee has slightly higher caffeine content compared to tea sample.
In this research, the high-performance liquid chromatography HPLC method using the HPLC model 1514 from Shimadzu Corporation was used to determine the caffeine content in tea and coffee samples. This method demonstrates excellent accuracy and sensitivity in identifying caffeine. Also, we get to find the qualitative and quantitative analysis of caffeine content in the samples. The qualitative analysis aimed to identify the presence of caffeine in both tea and coffee samples. By comparing the retention times of both samples under the identical chromatogram, it was observed that the retention time for both samples was same. This confirms that caffeine is present in both tea and coffee samples, as it eluted at the same retention time, which indicates that caffeine behaves similarly in both matrices. In the meantime, quantitative analysis involved in determining the precise caffeine concentration in the tea and coffee samples. Using a standard calibration curve, the caffeine content was quantified. The results show a slight difference in concentration between the two samples, with tea containing less caffeine than coffee. This difference could be attributed to the plant composition or the sample preparation steps, because both samples were processed similarly. Despite the slight difference, the caffeine quantification was accurate and reliable. This shows about the precision and sensitivity of the HPLC method. Next, the results indicate that both tea and coffee samples were successfully analyzed using HPLC with caffeine detected in both matrices. The matching retention time suggests that neither the tea nor the coffee matrix interfered significantly with caffeine separation, further confirming the method's accuracy and reliability. The difference in peak areas between the two samples correspond to their different caffeine concentrations, with coffee having slightly higher caffeine content than tea. In conclusion, the HPLC method proved to be effective in both the qualitative and quantitative analysis of caffeine content in tea and coffee samples. The HPLC method showed a high degree of precision and sensitivity, with low retention times that allow for high throughput analysis. This makes the method suitable not only for analyzing caffeine content in tea and coffee, but also for other beverages such as soft drinks, energy drinks, and kombucha in future research.
This study demonstrates the robustness and efficiency of high-performance liquid chromatography (HPLC) in the quantification of caffeine in tea and coffee sample. The validated method shows consistent caffeine elution with no significant metric interference from tea or coffee component, ensuring precise separation and measurement with a retention time of 2.6 minutes and a linear calibration curve equal to 0.9928. HPLC provided rapid and reliable results. The caffeine concentration were determined to be 0.0699 mg per milliliter for coffee and 0.0697 mg per milliliter for tea, showcasing the consistency of the method across different metrics. Additionally, HPLC effectively separates caffeine from other interference substances such as a tannic acid, caffeic acid, and sugars, making it suitable for diverse food and quality application. HPLC high-performance liquid chromatography is a highly effective method for measuring caffeine because it is precise, fast, and can separate compounds like tannic acid, caffeic acid, and sugars. However, other tools can support or even replace it in certain cases.
For example, gas chromatography mass spectrometry (GCMS) is great for identifying volatile compounds and detecting impurities. Liquid chromatography mass spectrometry (LCMS) offers even better accuracy for advanced studies such as analyzing caffeine's interaction in tea and coffee. For simpler tasks, UV spectrophotometry can quickly measure caffeine based on how it absorbs light, while Fourier transform infrared spectroscopy (FTIR) help identify specific chemical groups like sugar or acid. Other than that, nuclear magnetic resonance (NMR) spectroscopy, though less common for caffeine, provide detailed structural information. While capillary electrophoresis (CE) is a fast and eco-friendly option for analyzing compounds with low sample use. Using this tool along with HPLC can give a deeper and more reliable understanding of sample content and making them ideal for quality control and research in food and beverages.
HPLC is an acronym for High Performance Liquid Chromatography. The words chromato and graphy literally mean color writing. HPLC was pioneered by Friedrich Christian Schronben, 1799 to 1868, who began moving substances through filter paper, and Mikhail Shmoynovich Svet, 1872 to 1919, who separated plant pigments into calcium carbonate columns. High-performance liquid chromatography is the separation of compounds on a stationary phase column. The basic components of an HPLC system consists of 1. The mobile phase. This is the solvent or eluent that flows through the system and carries the liquid sample from the injector through the column to the detector. The solvent is contained in a reservoir at a higher elevation than the pump to maintain a positive head pressure. A solvent filter removes any particles that could potentially damage the system's sensitive components. 2. A pump. The first piece of hardware the mobile phase encounters is the pump. The pump pushes the mobile phase continually through the HPLC system at a specified flow rate. 3. An autosampler. After the pump, the mobile phase encounters the autosampler. An autosampler works by aspirating and injecting a precise sample volume into the mobile phase stream. The autosampler stores the sample vials or plates in a temperature-controlled environment and injects the sample when instructed by the data system. 4. A column. After sample injection, the mobile phase carries the sample to the stationary phase column, where physical separation of the compound occurs. Proper column temperature control is essential to conserving retention time precision, selectivity, and separation efficiency. 5. A detector. After elution from the column, the mobile phase transports the separated compounds, or analytes, to the detector. Most HPLC detectors work by converting a physiochemical property of an analyte into an electrical signal. And 6. A chromatography data system, CDS. The data system translates the signal from the detector into a chromatogram that provides qualitative and quantitative data about the sample. The data system allows complete control of the pump, autosampler, and detector. All of the instrument parameters, automated run sequences, and data collection are controlled by the data system.